Hello, everyone, and a very warm welcome to today's Spring into STEM lecture. This is our final lecture for medical physics and biomedical engineering in this series. And so what a fantastic topic to close out our series on. So before I hand over to Patrick, I just wanted to say if you have any questions for Patrick and the lecture, you can put them in the chat or the Q&A box and we'll come to them at the end of the presentation. So without further ado, I'd like to hand over to Patrick to take things away.
Hello and welcome to this final Spring into STEM talk. I'll jump right in. My name is Patrick and we're going to take a look at what brains are made of. So most of you, I'm sure, have seen a brain before, and if this were an in-person talk, I'd ask a show of hands to see who's seen an animal or human brain before. In case you haven't, it looks a little bit like this, but it'll be more gray or clay-like on the outside. And that is hundreds of years ago how people were actually investigating this question, trying to figure out what the brain is made up of. They took it outside of a diseased animal or a diseased person and just started cutting into it with a scalpel or a chain or a knife or anything. And that's also where the names come from of the major constituents: the white matter because it looks white, the gray matter because it looks gray, and then there's the cerebrospinal fluid in the center, and there's some other major areas like the brain stem that can be easily identified by their change in color. Now, as imaging methods got better and people started to cut the brain into thin slices and put them under microscopes, and they saw that it's comprised of many more smaller parts, most of which are neurons, and there's also glia, which are non-active cells, and these neurons are connected with axons, and there's blood vessels all throughout the brain to supply all of these cells with oxygen and nutrients. And these cells can communicate, which obviously when first imaging them, people couldn't see, but people could figure out later down the line. And if you zoom in even further, you will see that these cells are made up of molecules, proteins, atoms. There's a lot of water in there, as there is in the rest of the body. But unfortunately, all of these investigations into what the brain is made up of were performed on deceased individuals. Only very rarely would people actually saw open a skull and prod around in a live brain. And of course, that is because it's very difficult to image the brain without taking it out of a human. Now, over the last 60 to 80 years, there's a lot of new methods have been developed to image inside the human body. And with these, we can answer much more complicated questions about what the brain is made up of, aside from listing the types of molecules that the brain is made up of, but really looking at answering questions like, how does the brain change from children to adults to elderly people with neurodegenerative diseases? Why does it change the way it does? We see changes in people that do sports with high impact, like boxing or American football, where that leads to brain structure changes. And to really investigate how these impacts shape the brain or change some materials or constituents of the brain, we need to be able to image it while people are living. As I started off, microscopy is one way of looking at tissue, but typically it's very difficult to do so, especially at high resolutions on living tissue. Here you see a cell being imaged with a microscope. Now, in a clinical setting, imaging living tissue, ultrasound is a very easy and cheap way of doing so. Unfortunately for the brain, ultrasound doesn't really work because ultrasound does not go through bone. It'll just reflect on bone. So for imaging a baby in the belly of an expectant mother is easy because there's no bones in the way. But imaging the brain is impossible from a very early age once the skull fuses and closes up. There is an imaging methodology, however, that can look through bone, and that's X-ray. And here you see an X-ray image of a knee. X-ray radiation goes straight through bone or is stopped only slightly by bone. And that gives a very good contrast in the bone. So we can see there's bone and then there's tissue around it. Unfortunately, the tissue around it doesn't have a very good contrast. So there's muscles here, there's fat, there's tendons, there's skin, and you can see some slight changes, but it's very difficult to tell which is which and where they are. And it's even more apparent if you look at a CT image, is computational tomography, which is a 3D X-ray technique. The skull is very easily seen, but the inside the head, the contrast is very weak. So it's very difficult to discern the different tissue types and constituents of the brain. Magnetic resonance imaging, however, which I'm working in, is great at soft tissue contrast. On the flip side of the coin, it is much more difficult to image bone with MRI, and it typically takes a lot longer to get an image than with X-ray imaging. But it does have great soft tissue contrast, as you can see from a brain image here, where the cerebrospinal fluid is black. The white matter is aptly white, and the gray matter is a grayish shade on the outside. So it's very easy to see these different parts of the brain in an image. Unfortunately, one of the downsides of typical MR imaging is that it's qualitative. That means you can see the different contrasts, but I can't give you a value. So I can't say the white matter has a signal intensity of 10, the gray matter is 5, and the cerebrospinal fluid is 1. Or I can give you values, but they're relatively meaningless in the sense that they're not related to any specific physical quantity. And from the image, they're rather arbitrary. Another example of qualitative versus quantitative is in this race car. When I show you this image, you can answer the question, is it moving or is it stationary? With a high probability, you can say, it's probably moving because the background is blurred. But then if you try to answer the question, how fast is it moving? Or even more complicated questions, such as, is it accelerating or decelerating at this point in time? Those questions become impossible to answer from a photograph alone. So it's a qualitative photograph in that you can tell whether or not it's stationary or moving, but you can't say anything quantitative about its motion. If instead of a photograph, I would show a movie here, and the movie would track the car along a known stretch of road, then you could say, time the car on that stretch of road, and based on the amount of distance traveled over the time taken, you could then from the movie infer a quantitative metric of the speed of the car, or the average speed over that time interval. So if you have a movie, you can actually reconstruct quantitative parameters, which you couldn't get from the photograph. And it's pretty much the same thing for MRI. In a clinical setting, people don't have the time to take a movie, but they will rather take a picture, which gives them a qualitative insight into whatever tissue they're interested in. And they can often already say something about whether or not it's healthy or diseased, but they don't take the time to take a full movie and then do the corresponding reconstruction necessary to get the actual value of how diseased is the tissue, or what is it actually specifically made up of. And nowadays, there's a lot of people working on introducing more quantitative measures in MRI, and also making these quantitative measures more relevant in a clinical setting. And some examples that we have of these in brain imaging specifically is, to start off with, the structural imaging you've seen before. There are quantitative metrics that can be inferred from these, such as tissue density. or relaxation parameters that you can get in a structural sense. Diffusion imaging, which in my opinion gets the most interesting or fascinating looking 3D images, which basically is a way of measuring the connections within the brain. So it shows you which areas of the brain are connected with which, as essentially, as they're called, tracts that connect different areas of the brain. Functional imaging can tell us something about which areas of the brain are using a larger amount of oxygen than other areas, or than that area would be when there is no function. So that's a way to measure activity in the brain. Then there's blood flow imaging, which gives a measure of, as you'd imagine, the flow of blood in the brain, and the electromagnetic properties, which is the primary interest of my research in the brain. And the ones I show here is the susceptibility or permeability of tissue and the conductivity of tissue. Before we'll get into how to actually image these electromagnetic properties, I'll give a quick rundown of what they are and why we're interested in them. So the permeability, which is often referred to as magnetic susceptibility in MRI, essentially deals with how the tissue or material you're interested in reacts to an applied magnetic field. So in the case of a compass, it's very simple. If you apply a magnetic field through, for example, an actual magnet, I'm sure all of you have seen this before, the arrow on the compass will point in the direction the magnet makes it point. And that's because it's made of an iron or material that is very susceptible to this magnetic field. And likewise, you have materials which are not susceptible or less susceptible to the magnetic field, or react in an opposing manner to the magnetic field. And some of the examples I have in the brain is the blood oxygenation, which is also used to do functional imaging, which changes the susceptibility of the material to the applied magnetic field. Then there's iron contents, which seem to turn up a lot in dementia and other neurodegenerative diseases. And for example, calcium, which is in calcifications in the brain in elderly, which all have various impacts on the susceptibility of the brain. Then there's conductivity, which clearly tells you how conductive the material is. And you can imagine, for example, cerebral spinal fluid, which is essentially water. If you add salt to water, it becomes more conductive. And the same goes for a lot of the tissue in the brain. If there's more ions present, or for example, sodium, then the conductivity changes. And there are some very large changes in conductivity from cancerous tissue versus healthy tissue, as well as the conductivity of various cell membranes is differently influenced by this. So all of these are metrics we can infer from conductivity. And finally, the permittivity is the mirror of the permeability, but then for the electric field, as opposed to the magnetic field. So it talks about how material reacts to an applied electric field. And one example, which I'm sure you're familiar with, is a prism, which actually slows down some frequencies of light more than others. And that leads to a very nice rainbow. And that has to do because the prism has a different permittivity from the air around it. And in the brain, that's water content and also salt levels in the brain. Now, how do we image these? In MRI, all of our signal comes from hydrogen atoms and the spin in hydrogen atoms. And to image these, we apply a big magnetic field, which is why the scanner is so massive, to generate a very strong uniform magnetic field in one direction that aligns these hydrogen spins. And as I mentioned, the magnetic susceptibility influences the local field. So it changes how that field is perceived. And these changes actually propagate into the phase of your measurement. So we can use the phase to reconstruct that susceptibility. Now, for the electric properties, to actually get a signal, you need to excite these hydrogen spins again from their aligned positions perpendicular out of alignment. And when they relax back into alignment, that is when you achieve a signal in MRI. Now, this excitation flips the spins, and this flip is not uniform precisely because the tissue has electromagnetic or electric tissue properties, so conductivity and permittivity. And we can measure that flip angle, which will tell us something about them, as well as a phase offset, which may be difficult to see on the screen, but there is a slight gradient here because of these electric tissue properties. And with these measurements, we essentially reconstruct these quantitative tissue properties and then use those to be able to say more about what the brain is exactly made up of at various stages in life or during various activities. Unfortunately, this is much like the race car, an inverse problem, which is not as simple as reading off the values from your measurement, but it requires inverting the model that relates them to the measurements. And that requires a bit of complicated mathematics. But I think that's also what makes it a very interesting problem to work on, because it's not easy to do. And a lot of people with me are trying to make better reconstructions as well as enable faster imaging of these parameters, so that eventually this can just be incorporated in a typical clinical scan, and we get quantitative measurements for various diseases to be able to track the disease progression and the effectiveness of treatments, and in the end, improve healthcare. So with that, I'd like to thank you for your attention, and I'd like to thank the group I work here at UCL for their assistance and their inspiration. Thank you.
Thank you so much, Patrick. If anyone has any questions, please do pop them in the chat or the Q&A box. We've had a question about accessing the recording of this talk. So we are recording. It will be available on our YouTube channel and also the faculty, engineering faculty's YouTube channel. We just need to do some post-production kind of editing to make sure the captions are correct. So there might be a 1 or 2 week delay before it being uploaded onto our YouTube channel, but we'll be notifying everybody who's attended or registered that the webinar is available. So Patrick, could you maybe give us a bit of information about the research projects that you're working on, where you're applying this kind of the sort of MRI scans?
Yeah, of course. One of the projects I'm working on is currently imaging these properties specifically for Alzheimer's research. So we know that people with Alzheimer's have more iron in certain areas of the brain. But what we're trying to figure out is whether we can use, for example, these iron accumulations, as well as other physiological changes in the brain, like the networks of the brain, to inform us whether someone is at an early stage of Alzheimer's or what type of Alzheimer's someone has, as well as helping us track the disease progression, especially under the influence of treatment plans, where you could say, is the treatment actually effective? Does it actually reduce or slow down the progression we're seeing, or perhaps even reverse it? Which would, of course, be wonderful if we could reverse dementia in old people. Now, to be able to do that, the main thing I'm working on right now is trying to make the sequence faster. So we have ways of measuring these properties in a nice way, but it takes half an hour, 45 minutes, and you can't just put patients in the scanner for those lengths of time, especially not patients with far progressed neurodegenerative diseases. They may be confused, they may start moving. There's a lot of other issues with that, as well as if you want to be able to scan everyone who's even at risk of the disease or has early symptoms, it just makes more sense to be able to do so efficiently. So that's why we're currently working on speeding that up, trying to get it enough information in 5 minutes to be able to reconstruct these properties. But it's like shortening the movie you had to just a couple of frames, or taking less images of the race car, and still try to be able to get an accurate measure of the speed, which is of course easier if you have more information.
And what would you say is like your biggest challenge in carrying out your research into Alzheimer's?
Biggest challenge is a tricky one. There's a lot of small challenges that each make compound essentially. But I would say one of the bigger challenges is actually acquiring the patient data. We're not there yet in the project, but I'm foreseeing that will be a big step, because it requires scanning as many patients as we can scan, but also at various progressions of Alzheimer's, and obviously that's a big undertaking. Even if we have a sequence that's fast and efficient and we can just give it to radiologists and say, please scan them this way, I'm sure there's going to be a lot of issues that will pop up that we haven't thought of beforehand, because while we're working on this, we're scanning our colleagues or healthy volunteers, which are typically not elderly and Alzheimer's patients, because it's easier to scan those. But it also means that once you get to actually scanning the patients, there's always things bound to crop up which you haven't thought of, or you didn't run into while trying to improve your imaging.
And what other medical conditions do you like? Is the MRI technology particularly useful for spotting?
So in the brain specifically, there's things like epilepsy and lesions, or aneurysms, which are bleeds in the brain, which can be imaged in a very good way using magnetic susceptibility. But throughout the body, there are many applications also for these electromagnetic properties. To talk, for example, in the liver, liver iron accumulations, or alcohol-based liver diseases, that they can actually help investigate the deterioration in the liver. And there's one of my colleagues works on prostate imaging susceptibility, for example, for various types of, I think, prostate cancers. So I would say it depends. I think there's a lot of physiological processes as well in the body where having these quantitative metrics are very interesting. So for example, in athletes, sodium imaging is used to see whether ligaments are at risk of tearing or breaking when they put too much strain on them. But a lot of this is research that's ongoing, and there's, as I showed, there are many different quantitative parameters that can be investigated using MRI, and each one of those has different challenges, but also different opportunities when it comes to learning about the human body as well as treating diseases.
Fantastic, thank you. So we've got a comment. As they say that they don't have a question, but they wanted to say that they love the race car analogy. It really made it make sense. So that's fantastic. If anyone else had any questions, please do put them in the chat or the Q&A. Let me just see. Doesn't look like we've got anything else coming through. So if there's no other questions, I'd just like to say thank you so much, Patrick. That was a really, really fascinating lecture. And also, sorry, we've got one more question. What's your favorite part of working in MRI?
I'd have to say that's the opportunities, because a lot of people in our department work in labs, and they have their detectors and they do their experiments. And in my case, My lab is in a hospital far away. With a scanner, I get access to maybe a couple of hours a week. But the marvelous engineering that's gone into that scanner, and that's also why they're so expensive, allows for a lot of really cool tricks when you actually look at the physics of it and understand how really all of it works together. And I'm fascinated also at all of the wonderful tricks my colleagues think of to improve our imaging methods, to improve our opportunities with it. And every year I'm amazed at the new things people manage to do with scanners, which are sometimes 10, 15, 20 years old, or methods that are developed on newer scanners that can then be applied to older scanners as well. So it's really being able to play with this wonderful machine and use it to its fullest potential, or at least try. I think that's the cool thing about it.
That's fantastic. Thank you so much, Patrick. That was a really fascinating lecture, and it was a great way to close off Medical Physics Spring into STEM lecture series. And I'd just like to say thank you to everybody for attending, and also our other presenters for presenting throughout May. This is the 6th lecture that we've had from Medical Physics. I think there's another week or so of lectures taking place throughout the faculty, but this was a great way to close off our contribution. So thank you so much, Patrick, and thank you everybody for attending today's session.